James E. Watson was born on April 6, 1928 in Chicago, Illinois. He always loved birdwatching with his dad every Sunday and read many classic literature during his free time. He went to Horace Mann Grammar School and graduated from South Shore High School in 1943. Watson then earned a scholarship to go to University of Chicago and stayed for 4 years to earn a bachelor's degree in zoology. He then went to Indiana University where he studied genetics and finished with his doctorate in 1950. Then Watson worked as a National Research Council Fellow at the University of Copenhagen. This is where he was working with bacterial viruses and was trying to see what happened to their DNA when they were infected. In 1951, Watson followed one of his mentors to a zoological station in Italy, where he met Rosalind Franklin and Maurice Wilkins using X-rays to observe the diffraction pattern of DNA in crystal form. This helped spark an interest for Watson, so he started researching on the structural chemistry of nucleic acids.
Francis Crick was born on June 8, 1916 in Northampton, England. He finished his elementary education at Northampton Grammar School and went on to receive his Bachelor of Science at University College in London. After, he stayed in London to further his education for a PhD in physics. In 1939, his studies had to be halted as his country, England, went into World War II. He worked during the war at the British Admiralty and left there to go to the Strange-Wade Laboratory in 1947. Once there, he was displeased with his assigned research project, and with that he moved to Cavendish Laboratory. Crick then joined a group of researchers led by Max Perutz and John Kendrew and began to study the crystallographic protein structure.
In later months of 1951, Watson also went to Oxford University's Cavendish Laboratory in England for research. This is where he met Francis Crick and discovered that they both were interested in nucleic protein and acid. They then worked together and discovered the structure of DNA by 1953. James Watson and Francis Crick published their discovery in a newspaper called Nature in April and May of 1953. They said, "We wish to put forward a radically different structure for the salt of deoxyribose nucleic acid. This structure has two helical chains, each coiled around the same axis. The bases are on the inside of the helix and the phosphates on the outside. The novel feature of the structure is the manner in which the two chains are held together by the purine and pyrimidine bases." And in May 1953, they said, "To each sugar is attached a nitrogenous base, which can be of 4 different types. 2 of the possible bases, which are adenine and guanine, are purines, and the other 2, thymine and cytosine, are pyrimidines."
Where I had a big and a small molecule, and so how did you do it? Somehow you had to form link bonds. So here's A, and here's T, and I wanted this hydrogen to point directly at this nitrogen. So I had something like this. Oh. So then I went to the pair, and I wanted this nitrogen to point to this one. And I went like this. Whoa, they look the same. And you can put one right on top of the other.
Therefore, Watson and Crick explored the human genome, discovering that the structure is a double helix which is made up of phosphate and sugar chains linked by 4 chemical bases. Also, Watson and Crick found out that when DNA needed to be replicated, the 2 DNA strands first separated. The rungs of the ladder breaks apart and the result of each strand becomes a template strand in order for a new strand to be created. Then, each new strand bonds to its template strand, and the result is 2 new DNA molecules, each consisting of an old and new strand. This is called mitosis. As a result of their discovery, James Watson, Francis Crick, and Maurice Wilkins won the Nobel Prize in Physiology in 1962. James Watson made a speech at the Nobel Banquet in Stockholm on December 10th, and he said, "Our discovery was done using the methods of physics and chemistry to understand biology. I am a biologist, while my friends Maurice and Francis are physicists. I am very much the junior one, and my contribution to this work could have only happened with the help of Maurice and Francis. At that time, some biologists were not very sympathetic with us because we wanted to solve a biological truth by physical means. but fortunately some physicists thought that through using the techniques of physics and chemistry, a real contribution to biology could be made. However, there was a controversy about how Rosalind Franklin should have won the Nobel Prize with the others, since she helped with Maurice Wilkins in finding the X-ray display of DNA. However, some say that she did not fully help Watson and Crick with the discovery.
All of the things explored by Watson and Crick led to the exchange of ideas that eventually led to Beatles and Tatum's one gene, one enzyme hypothesis in 1941. They discovered how genes help direct the synthesis of enzymes that control the metabolism process. Therefore, geneticists are able to discover that DNA forms mRNA by transcription, mRNA direct the formation of the proteins by translation, allowing protein to express the trait of an organism. All of the exchange of ideas led to the development of biotechnology, which is genetic engineering or the manipulation of DNA. Scientists are now able to transfer genes from one organism to another. Biotechnology began to form during the 1960s when Werner Arbor, Daniel Nathans, and Hamilton O. Smith were able to discover restriction enzymes, which is now one of the most important tools in biotechnology today. This is how geneticists cut DNA and allow sticky ends from it to glue the different genes back together. Mixing genes allow different organisms to produce different proteins that they usually do not produce on their own. This also allows more medical applications such as better treatments or even some permanent cures for genetic diseases. Pharmaceutical products should also allow mass production of hormones. There could also be environmental cleanup such as genetically engineered bacteria that can digest oil. This eventually led to genetically modified organisms or GMO. For example, some plants are able to produce new proteins to help protect crops from insects such as Bt corn. The corn is able to produce a bacterial toxin that kills caterpillars and other insects that try to eat the corn. Restriction enzymes help with forensics, medical diagnosis, paternity, evolutionary relationships, and more today. Scientists are able to compare DNA fragments based on size, since all people have different DNA and therefore restriction enzymes cut up a sequence a different number of times in different people. Scientists are able to separate DNA fragments by using gel electrophoresis, since DNA is negatively charged and is able to move on an electric field. James Watson and Francis Crick's discovery was very important and necessary for the development for our society today. Their findings of the two strands of DNA led to more scientific research on the DNA, such as Werner Arbor, Daniel Nathans, and Hamilton O. Smith's discovery of restriction enzyme in order to further advance biology, which led to biotechnology. Therefore, genetically engineered food and organisms are created and forensics and paternity was able to be developed. Biotechnology has most importantly allowed the scientists to diagnose diseases and create treatments or drugs to cure them.
This wonderful symmetry which we saw, the morning of February 28th, 1953.
Without Watson and Crick's discoveries, many scientific feats would not have occurred.